Good morning to everyone. My name is Giacomo Baruzzo. I'm a researcher at the University of Padua. And in this talk, I'm going to show you, here we are, okay. So I'm going to show you our comprehensive benchmarking of method to identify differential abundant taxa in 16x data, a benchmarking that has also been recently published in PLOS Computational Biology. So each time we want to analyze the species in a sample and the modern ways to high throughput sequencing, we end up with the first step and we collect the sample, we perform sequencing, and when we have a first step of bioinformatics, when we move from reads up to what is called the abundance matrix. A table, a matrix, where on the row we have the taxa, on the column we have a different sample subject that we collect during the experiment, and this is really the starting point for the second part of the bioinformatics pipeline, when we, from abundance data, we can perform many downstream analyses. The typical one, alpha diversity, beta diversity, but also a very standard and important analysis is what is defined as differential abundance, or just DA. Roughly speaking, a computational way to identify which taxa are significantly different between the two or more experimental conditions that may be present in our experiment. So the problem, if you are familiar with this task, is not the lack of method. The problem is the choice of a method. So we have many methods based on different modeling, having different assumptions. And so the problem is the choice. So which is the one that I should use? There is a best method for all datasets. There is a best method maybe for a specific type of dataset. And so this is really an issue. This is really a problem. But for this reason, in the past, many study has been performed to help the final user in identifying which is the best method to use. So we are not the first one. There is considerable literature, many amazing studies that try to answer the question, which is the best method, or more precise, which is the best method for the characteristics of the data. So these are just the 5 most recent one and main one. But as you know, this is true for metagenomics, this is true for all bioinformatics. It's a very fast evolving field. We have new method that comes out each month, each year. In the last 2 or 3 years, there is a huge also effort in developing the first reliable and validated simulator for count data in the metagenomic field. I'm thinking about Metasparsim, Spardosa, so the first ever simulator to generate in a realistic way count data from 16S or shotgun technology. So we have more reliable way to even generate ground truth data. Then there is a lot of covariates of interest to study: the sample size, the number of differential abundance data. So there is many things that we want to study. Also, the combined effect, how they affect the performance of the method. And so at the end, there are many scenarios and many metrics that we need to use to cover the complete performance of method. For this reason, we proposed a new benchmarking study, an update in this time series of study, where we consider even the last recently developed method for differential abundance analysis. We use state-of-the-art simulation techniques to add realistic ground truth data to be used during the benchmarking. And we study the different covariates and their combinatorial effects, so more than one covariates tuned at the same time, at the end generating many scenarios and assessing them with many metrics. So, which is the overall framework of our study? Well, as in any benchmarking framework, you start with having some ground truth, so the label that you can use to then assess the performance of the method. In terms of simulation framework, well, you end up having 2 main actors: a simulated abundance matrix and the corresponding ground truth knowledge about which taxa were generated as differentially abundant in the corresponding abundance matrix. Then the abundance matrix becomes the input of a differential abundance tool that you want to assess. You run the tool, you collect the prediction, so which taxa they define as being differentially expressed, and then you compare the results. So this is, if you want, a very straightforward way, is the typical workflow in all the benchmarking study, and so the workflow that even adopted in our study. Once you have set up this workflow, well, it's just a matter of choosing which method you want to test. And in this case, we try to cover the widest set of methods possible. So we start from methods specifically developed for metagenomic data. You can recognize probably ALDEX, eBay, Yancom, and so on. We bring method very common in other fields. For example, bulk RNA-seq, you probably recognize the seq and the jar, as well as some baseline method that's just statistical text like the Wilcoxon, and it's a modified version of it when we use data that are not count data, but they are use the CLR transformation, so we try to cover the compositional bias. So this is the set of methods that we tested, 13 methods. What about the metrics? Well, we have a set of ground truth label, the infinite ground truth label. We use the typical metrics to analyze that you can use even to assess machine learning predictions. So false positive rate, full discovery rate, recall, and many other metrics. What about the simulation framework, roughly speaking? How we obtain the simulated abundance metrics and the ground truth taxa in a realistic way, because if data are realistic, then your results are useful. Otherwise, you're generating garbage. So if you're familiar with the problem of simulating or even analyzing 16S count data, you know that data are quite messy to analyze. A lot of zero, a lot of noise, both technical noise and biological variability. The data are compositional. They rely on the simplex, not on the Euclidean space. They are heteroscedastic. The distribution are high left skew among towards very low abundance taxa. So there is a lot of technical methodological issue that makes the analysis of this data challenging, and also their simulation. For this reason, different from some of the previous study, we don't develop our ad hoc simulation procedure, but we start using the available count data simulator that comes out in the last year. In particular, we use metasparsing. Spoiler, we are also some of the author of metasparsing, which is the first, to best of our knowledge, published count data simulator for 16S data. And it uses a gamma multivariate hypergeometric model to model both the biological variability and the technical variability typical of all 16S data. And also, it takes care of simulating in a realistic way all the characteristics that you see here on the left, the compositionality, the heteroscedastic, and so on. Metasparsing was designed to just simulate a different group of sample, so not just two groups that have some differences. And so we extend the framework to allow us, starting from one experimental group, simulate a second one with the exact characteristics that we want, that we will assess during the assessment. To make the things as realistic as possible, we start from 5 very different real datasets that cover different biological aspects, but also technical aspects. You can see different technology, different 16S region, different ecological decay. And from these 5 datasets, we estimate their parameter. The parameter then be used to make 5 main simulations in our benchmarking study to cover, again, the widest range possible of technical and biological aspects. Once we have the 5 simulation parameter to have this 5 flow of simulation, let's say, then we start to play with covariates that are at the end the things that make one dataset different from the other, the number of sample, the amount of differential abundance taxa. In particular, we make other simulations starting from the 5 initial per set of parameters that, for example, study different the availability or not of differential abundance taxa. So we create a scenario that is very useful to identify type of 1 error for the method. We simulate no taxa to be differentially expressed. We also change the percentage of taxa, 5, 15, and 20% that are differentially abundant to see if the performance of the method are affected by this element. We also study the presence or absence of differential abundance taxa at low abundance, in the low abundance range, because as you can imagine, identify taxa that are differentially expressed at low abundance is another problem compared to high expression level. And also we play with the biological ability, increasing or decreasing to see how this affects the performance of the method. In terms of the technical aspects, well, we study the effect of using a specific normalization compared to the default one suggested by the author of the tool, the effect of a sequencing depth, increasing, decreasing the sequencing depth, or for example, having an unbalanced situation, having condition 1 with 2 times the read of condition 2. And also playing with the sample size, the number of samples that we have in each experimental condition, 10, 25, 50, and 100. So 4 different scenarios. So if you consider the 5 initial data sets which estimate the parameter, all of these are other simulation scenario that we could create. And we test in a combinatorial way. Well, if you did a little bit of math, 5 initial scenario times all the covariate and the combined effect, and to be robust in a statistical way, each thing was repeated 50 times. At the end, we simulate 12K almost matrices that were fed as input to the 13 methods. As you can imagine, a lot of time on the department cluster. So the assessment is quite rich. All the covariates were inspected. So we have just 5 minutes left. Let me just give you a very quick overview of the results. They are all on the paper, but just some hint. Well, here is just for one of the scenario, the different sample size here, the different percentage of the taxa here, and you see the mean FDR on the X, the mean recall on the Y-axis. And you see that overall, the method have typically a good control of FDR, good recall, but these two things are true when the sample size is high. So I choose this among all the possible results because this is one of the major take-home message. The performance of the tool are good when there is high sample size. At low sample size, come on, man, we are here. So, and this is not just only result. If you take the previous benchmarking study, it's quite clear even there. So the number of sample is really the key factor for our field. Having a larger amount of sample is necessary for the tool that we are using nowadays. So more in general, you know, a lot of covariates, a lot of things. There is the performance of your tool considering that specific characteristics of the data. But when one of the reviewer was right, say, okay, try to summarize the results. Well, I know it's very useful. I hope a nice picture. It takes 2 days to do it, but hopefully provide a quite compact and a summary of the results. So what we did here, we consider 4 elements, 4 metrics: the precision, the recall, and the partial area under the PR curve, and the percentage of simulation which a method was able to identify at least one taxa as differentially abundant. We define different threshold for each of these 4 metrics. You see here is how much the pie chart is full from 0 up to full, black to white to black. And we sort the method for the best FDR. Since the main covariates that have the main effect is at the end of the sample size, you see here sample size 10, 25, 50, and 100. And the 5 columns are the 5 main initial data which estimate the parameter. What we see here, well, precision, a lot of black dot, meaning that the methods are typically good in controlling the FDR, having a good precision. Recall, well, as I told you, with few sample, the recall is typically not very good. We need to increase 25, 50, or even better, 100 to have an increasing recall, so have higher recall. And this is quite similar even for the 4th metric, so the area under the precision recall curve, and even for the ability to the tool to identify at least one differential abundant taxa. Here, all the dots are black. Here, half of them are white. So, to summarize more than 70 pages of article, the main results, last slide. Methods have typically a good control of type 1 error. At small sample size, majority of them are not able to find any differential abundance taxa. In terms of recall, that it seems the metrics that is harder for the method to achieve in a good way. FDR, precision is good, recall require a lot of sample. Effect on normalization, where the normalization that we use, GMPR, compared to the suggested one, have an effect only on the relative ranking of the method, meaning that with some normalization, method performance increase, but the relative ranking between the method remain the same. The library size in our test doesn't impact too much the performance. And if you want to just summarize everything, in terms of sorting method for FDR, it seems that we have low sample size. One of the best approach to use seems to be Maslin 2, because I have a good control of FDR and one of the highest recall, even if highest maybe is just 20%, but still better than the other. When we have higher sample size, then it seems that Aldex, X2, eBay, and Ancom seems to be good approaches. Take home message from this talk. So spend well your money. If you have more money and need to choose more sample or more read, go for more sample. I mean, I guess that the plot were quite clear. I mean, not always just a matter of money. Maybe you don't have the sample, but if you have these situations to choose how to spend your money, go for sample. Want to find the differential abundance bacteria at low expression? Well, cross your finger, because there is a lot of noise there, a lot of zero, a huge variability. It's really an hard task. It's not method fault, probably. It's just a very hard task. To analysts, if you need to spend some time to tuning your pipeline, well, spend more times in choosing the right method, for example, compared to choosing the right normalization, because it has an effect, but it's not a significant one. And for developers, if you want to update your method or develop a new method, well, precision seems to be already quite controlled by current techniques. The recall is the main issue. So all I have explained in this talk is available in this article, that we would consider not only a benchmarking study, but more a benchmarking framework. We release the package that we use to perform the simulation and the assessment. All the data, input and output, are on Zenodo, and everything, to make easy to use, is on Docker. So we would like that this work will be a building blocks for even maybe for future benchmarking studies. So everything is here. Let me conclude this presentation saying thanks to Barbara, the last author and the PI of our group, and really Marco, the guy there, because I'm just the one that present Marco work. So Marco is the first author. This is his PhD thesis, but unfortunately he was not able to be here. So I'm just the guy that is taking credit for Marco work. So this slide is to say all you see is Marco effort, not mine. I'm just co-supervise his thesis. And obviously thanks to all the research group. Thank you for your attention.
Yeah, time for two maybe very quick questions.
Thank you. Just wondering how you deal with some kind of double dipping in this work. I think the DA method heavily depends on the assumption. For example, LDX2, which assumes that it's a multinomial Dirichlet distribution in the microbiome data. DC2, this is a negative binomial. And when you do your simulation, you assume that it's gamma multivariate distribution. Yeah. And I assume maybe if there is one recent paper about the gamma Poisson distribution for the DA method. And I think maybe if you use this method on your simulation data, it will perform best. And it is more like a double dipping question. That is maybe what a method based on the simulation, what's the method for your DA test? That is to be match or not match. How do you overcome this one as in the morning keystone test, keystone stage?
So yeah, across all the 13 methods, no one has a modeling that match the model used to simulate the data.
There's a reason why that already use this method, which is gamma Poisson distribution, very similar to this one in Java.
Yeah, I mean, if you are using the same model to generate and then to assess the data, is a cyclical augmentation if you want. So it could be challenging. We just avoid this issue using something that was not used. But to answer to your question, if you maybe believe in that model and you want to use to generate the data, I guess that the best things that you can do is to create a lot of different simulation scenario covariates. So bring that stuff to the extreme case where maybe I don't know, a lot of different differential abundance feature compared to less. Maybe even if a model is the same, you are creating an emerging behavior from the other covariates that you plug in the data that maybe can alter something. But I mean, is by definition a cyclic argumentation. So I think that the best you can is just play a lot with all the covariates that you can try to plug in your data to make some emergent behavior maybe appear in the data.
Thanks a lot. Do you think your results will stand similar in the same trend for shotgun metagenomics, or would you expect because of the data structures, some of them recall will improve or get better?
I mean, the simulation framework that we designed and also the simulator that we used was tested and validated only of 16S data. We are aware of other people not in our group that using our simulator, even extend it to shotgun, and they said they are quite happy. But if you ask to us, we never try to extend by our own. And so, to be completely honest, I would not able to answer in a precise way to your question, because we just think about 16S data. There are groups that are extending it, and they seem happy, but nothing that I do with my hands, let's say.